All right. Let's give like a minute so that they are coming in. So while we're waiting for those who have been here already, so I want to make sure that everyone is aware that the talk is going to be recorded. And you have to, so if you have any question, feel free to put into the chat. What's going on, Callie?
Oh, you just got muted there for a second.
Is there something going on?
You're frozen there for a second.
Oh, I see. It's the internet. Yeah, so just sharing with those who are already here that please feel free to put the question into the chat if you have any question, and you're also welcome to just open your microphone, open your video, to talk with us as well. But keep in mind it's going to be recorded and you'll be shared on YouTube. So with that, me pull up the intro slide real quick.
Should I stop sharing?
Yes, that would be great, Callie.
Okay, I'll just briefly say, hi everybody, I'm Callie. I would really love to hear who's in the audience today. And I've got a lot of different components during this talk where I'll ask you questions. So if you want to use the chat to answer these questions, like what's your name, where are you from, and then what's a plant, animal, or microbe that represents your personality? I think that's a fun way to kind of get to know who's in the room. So anyway, if on the back burner, please do that.
So for the formal start, thanks everyone for joining us again, and also thanks Dr. Callie Chappell from Stanford who will be here with us today. So the talk title today is LABrary Biotechnology for Everyone, and Dr. Callie is like we met them at the Spirit of Asilomar conference just earlier this year. Both Kato and I went to that conference where Callie is one of the organizers, technically. I mean, or maybe like one of the lead next generation leaders, organizers who are helping us getting to know each other and also like trying to make impact to the bio community. So technically, Dr. Callie is a postdoctoral researcher at Stanford, and they had been working on a lot of projects, especially for trying to make the biotech accessible for everyone, and that includes all of you. So there's a lot of works around the science part where Callie has been working with both Brew Andy and Jen Brophy. Jen just like hopped onto our meeting just like 2 months ago. So, there are some connections right here. And Andrew is like very supportive of a lot of projects that we have been working on. And with that, I don't want to oversay it, but you see a lot of projects that is very reachable for all of you. I mean, if you're not from Africa, but from some space that may not have a lot of access to things, I think this is going to be a very interesting talk where you can take a lot of inspiration to work on some other things that might be doable in your space as well. So with that, I don't want to take too long, and I'm going to give the floor to Callie. So with that, thanks a lot again, Callie, for being with us today. I'm going to give the floor to you. And for everyone who is in the call, if you have any question, feel free to put into the chat, and maybe like use the hand raise and like try to, share with us what kind of question or what kind of enthusiasm you may have as well. I'll stop sharing right now. You can take it away.
Awesome. Well, hi, everybody. I'm so honored to be here. It's incredible what Kato and ICE and many others are doing with SynBio4ALL Africa. And when I got the invite to present here, I was so flattered and honored. So it's really my pleasure to be speaking with you today. And I really hope that today's presentation is more of a dialogue than just me talking. I really am dedicated to trying to make biology accessible for everyone. But I don't know how to do that. I don't know exactly where each of you are coming from, what resources you have access to or don't have access to, what you're excited about. And so for me, this is a really amazing opportunity to learn from you about what you're excited about and what you need, and see if I can help support that in any way, shape, or form that I can. So I'm going to go ahead and share my screen. Let's see here, and then we can get started. Okay, I'm going to share here and share sound. All right. All right, folks can see that?
Yes.
All right. Perfect. All right, let's go. So, I am Callie Chappell. I am a postdoctoral researcher at Stanford. I'm going to be presenting actually not on my current work, but actually prior work that I did while I was working with Drew Endy and David Kong at the MIT Media Lab. But the big question for this talk is, what is the future of synthetic biology? And I'm seeing people aren't really in the chat right now, but I'm really hoping that folks can get in the chat or just unmute themselves and share. I'm going to be posing this question for you in a minute. But as Ice mentioned, I wanted to kind of start off with a particular vision that was shared with me through the Spirit of Asilomar Next Generation Leaders Program. So as Ice was sharing, this was a conference that kind of gathered together many leaders in biotechnology earlier this year, and really thinking about what the future of biotechnology can be. And I had the great privilege of working with a really amazing cohort of about 50 early and mid-career researchers from around the world in envisioning this, the Next Generation Leaders Program. And I wanted to highlight a vision shared by Kajelcha, who's here actually on the call today, about what he saw as the future of biotechnology and how the rest of this talk will be an attempt to try to really carry that forward.
My name is Kajelcha from Addis Ababa, Ethiopia. Biotechnology is one of the most transformative technologies of our era and must benefit all, not just privileged few. The action I want to see is a worldwide investment in fair access to biotechnology, particularly in low and resource-limited countries. This can be achieved through improved research collaborations, open science efforts, and localized biomanufacturing. Thank you so much.
Thanks, Kajelcha. Maybe Kajelcha will put in the chat too, more about that. But I was like, yes, we need to have decentralized biomanufacturing. We need to have local labs. We need to have open, accessible science. And really trying to carry forward visions like this is the point of this talk. And so now I want to ask you the same question. What do you see as the future of synthetic biology? What are things that you're excited about or want to see? You can put in the chat, or you can also raise your hand. But we'll just take a pause here really quick, and I'd love to hear from you.
I'll start as an icebreaker because, my name is Ice anyway. Why not just break some ice? Yeah, so I think like just like everyone here, we want the future of the SynBio to be more of the accessible discipline. Like we know that a lot of the products of the field itself has immersed into your everyday life already, and I want people to realize that more of like what kind of transformative tools and technology has getting close to everyone. So in my opinion, I want to make sure that like not just the educational part that everyone is aware of what is going on, but everyone can access and try to see what they can do on the end to make this useful as well. And for others, if you are not in the situation where you can unmute or speak up easily, you can just feel free to type into the chat.
Yeah, Emma, do you have a vision for the future of synthetic biology that you would like to share?
Okay, I would share what I think as other participants are thinking about what share. The future of synthetic biology, I see it as helping to solve the current global challenges, more especially like climate change challenges, then challenges in toxicity, then challenges in like ecological challenges, whereby we are having like maybe biodiversity being distracted. So I see synthetic biology as a solution in future. And I see it as number one solution to the grand challenges which are being faced by the world currently. Yeah.
Yeah, thank you for that, Emma. We're going to talk about kind of the environmental side of SynBio, and that's personally what I research, not in the work that I'm presenting here today, but I'm very interested in that, those two questions of accessibility and the environment. Do we have anyone else that wants to share? Thank you, LABrary, for putting in some information in the chat. Okay, I'm hearing improved access and applicability to be able to address most of the problems being encountered. Yes, absolutely. And I feel like it's like folks like me, honestly, that are not always the most equipped to actually ensure that things are applicable. Try to design for the context that we're familiar with, but all of us only have experiences in our own, like only have expertise in our own lived experience in many cases. And so thinking about how increasing accessibility to doing SynBio can make sure that the things that we're creating are not just accessible, but also applicable in more contexts is something that I'm trying to carry forward, that you'll hear about in this talk. Okay. Do we want to hear? I don't know if people are in places where they can share, but I want to make this a more engaged discussion. So if you find yourself moving into a situation where you can turn on your camera or you can unmute yourself or you want to add in the chat, I really welcome that. But really looking forward to hearing more. Oh, we've got another one. SynBio is going to be part of the research solution. This is an interesting creative tool. I think so too. It's so amazing that we can work with life in this new way for creative solutions. And so I'm really interested in hearing what sorts of ideas y'all have for creative solutions with synthetic biology. So what I'm hearing from folks is that everyone wants biology for everyone, which is the name of the research program that I run at Stanford. And specifically, given the audience here, synthetic biology for everyone. And so how do we get there? Unfortunately, I'm not going to be able to give you all of the answers, but I'm gonna share a couple of things that I've worked on here and hopefully engage you in conversation about what you're hoping to push forward and create yourselves. Oh, I'm seeing another one. As a health biotechnology researcher, I believe that synthetic biology will enable engineered microbes and cells to produce novel therapeutics, diagnose disease with precision, and create personalized regenerative medicines, fundamentally transforming therapeutic development and healthcare delivery. Yes, I don't work so much in the healthcare realm, but I think that this is a super important area that by enabling more people to engage in synthetic biology from different places, different positionalities, and different ways of knowing, that we can create better therapeutics in healthcare and health approaches for folks around the world. So today in this talk, I'm going to kind of tell three different stories. The first is kind of reimagining what science and biotechnology is through the lens of art and culture. The second is thinking about education and specifically reframing education biotechnology as a way to center power in local knowledge as opposed to being like, I'm an expert, I'm going to tell you what you need to know, right? How do we amplify and celebrate knowledge that already exists in communities? And then the third is to empower communities through creating new local infrastructure that makes synthetic biology and biotechnology more accessible. So in this first story about reimagining science through art and culture, I really want to make the argument that art can reimagine science. And I just wanted to share a little bit about who I am and where I come from. I grew up in the United States, in a pretty rural part of the United States. This is a picture of like above my hometown. So you can see there's beautiful lakes, there's a little island there, there's forests, there's a lot of farmland. And while the local ecosystem and ecological community was very rich, there wasn't a lot of access to science. And I didn't grow up knowing anyone who identified as a scientist, and I didn't really feel like science was for me. But I really grew to love what I later learned to understand to be biology. I really learned to love those forests and the water and the animals that were around me through art. I had artists in my family and I had artists around me, and those people taught me to look at the dew on a fiddle leaf fern or be curious about why one tree or one plant was growing one place rather than another, and to really draw that. And that curiosity that was sparked through art really later got me interested in pursuing science as a discipline. And so I was always that little kid who loved to draw, and I'm curious if other people here like to draw or have artistic, creative endeavors. Oh, we've got Diego. Hi, Diego. But I kind of did that through doing scientific illustration. So you can see here some mushroom pictures that I drew. And being an artist really taught me how to look at the world scientifically. And so later I became a biologist. I was studying biology professionally, but I didn't really understand, even though art got me interested in science, how it could relate to science as a practice. And so then I was like, you know what, I could actually like publish this stuff. So I was starting to do drawings and illustrations, and I became a professional scientific illustrator in addition to being a scientist. And so this kind of, I think, is a trajectory that many people who are creative in science fall into, which is, hey, I'm a creative person. How does science connect to society? Well, maybe art can communicate science to society. Maybe art can make science more accessible so more people can be engaged. And while I think that that's really important, I think there's this relationship between science and society doesn't just go one way, from scientists communicating their findings to society, but the other way as well, that shows that society actually influences how science operates. And art can be a bridge that shows that science is actually embedded in society. And so I think that that's a really important point when thinking about how equity and access ties to biotechnology and synthetic biology, that it's not just about communicating the science to more people. It's actually about changing how society operates and how society influences how science operates to actually create structural changes to how more people can be engaged in synthetic biology and actually shape the future of what synthetic biology looks like. And so to give an example about this, I wanted to share a little bit about a recent gallery exhibition that I did called Indigo. That was part of a group show, Emergence Art from Life, that was in Los Angeles late last year. This was in partnership with Dr. Nasa Senate Armstrong, an assistant professor at the Fred Hutch Cancer Center. And the focus of this exhibition was highlighting this dye, so a chemical called indigoidine. And indigoidine is an indigo-like dye produced by a bioengineered yeast. So a group of researchers created this rhodotorula yeast that takes in L-glutamine and turns it into indigoidine, which is this bright blue dye. And we were thinking about indigoidine and indigo in the context of the global tradition of indigo dyeing practices. So the indigofera plant produces indigo dye. Here's an example of indigo dyed cotton from Nigeria in the 1960s. But there are a lot of different practices of indigo dyeing around the world. But there was a time when in Europe, for example, European colonizers really wanted to have indigo dye to fuel a lot of their trading practices and established, many folks here may know, like colonial dye production, for example, in British colonies in India and in the American South and plantations. And so there was this history of colonial exploitation for indigo that was related to producing the indigo plant, even though that practice originally was rooted in many communities around the world. And so later, instead of having to produce indigo from plants, there was a chemical synthesis of indigo. And so now a lot of the indigo used to dye jeans is through this chemical synthesis process, but has a huge environmental impact, particularly in the global south, associated with the textiles industry. And so the piece that we were developing was thinking about this colonial history and neo-colonial history of indigo production and asking, will synthetic biology be different? If we have a world where we can biomanufacture indigo, will that biomanufacturing process continue to have a legacy of the same colonial exploitation that indigo production has historically? Or is there an opportunity for it to be different? And so I want to highlight one way of thinking about community-based bioproduction. This is something that Kajalcha talked about in the initial vision that we shared at the beginning, which is that local communities control and benefit from synthetic biology products as opposed to having that knowledge be exploited and moved to other parts of the world. And so I want to highlight work done by Benno Juarez, who runs a floating fab lab in the Amazon. And you can see this is some of their stops here in the Amazon. And it's actually on a boat. And what Benno does, he works with local communities to do biotechnology development that's really focused on local knowledge. And one of the things that he did was actually create a local bioreactor. And so this is a mock-up of the bioreactor. Here's a picture of Benno actually holding the bioreactor. And when I asked him, what is your vision for the future of biotechnology? He said, I envision a world where biotech inspired in ancestral knowledge becomes a force to switch from a predatory to a regenerative culture, both ecologically and socially. And I think that that is really aligned with a lot of the visions about biotechnology that have already been shared shared here. And so really we can use conversations about science and society with art as a jumping off point and culture as a jumping off point to really imagine different futures for biotechnology and ones that are aligned with the visions that folks have shared here in Benno, right, where ancestral knowledge, cultural knowledge, and community really is the driver of innovations in synthetic biology. So, okay, how do we get there? What's the next step? So the next part of this talk is really thinking about education. So how do we center that power in local knowledge? How do we change the trajectory of synthetic biology to be rooted in cultural and local and in the voices of young people? And to answer this question, we really wanted to ask ourselves, well, who should be the leaders in biotechnology? And sitting down and through many, many, many mentors and collaborators of mine, we really said that we think that youth, that workers, that oppressed communities or communities that don't have access to technology should really be the leaders. People who will be the most impacted potentially negatively by biotechnology should really be the ones at the forefront of designing and developing biotechnologies. And that universities and elite institutions and in the global north should be in listening and following mode to what those visions of others have. And so in order to do that, we really were imagining how we might expand the types of knowledge we consider to be biotechnology through art, culture, and community as that kind of accessible and societally centered entry point to thinking about biotechnology development. And so the story I'm going to tell you about for this section is related to an educational program that I've been involved in for many years called BioJam Camp. And before I tell you about BioJam, I want to tell you about the story of BioJam. So BioJam was co-founded by Dr. Rolando Perez, who's a bioengineer at Stanford, and Corinne Okada-Takara, who's a really incredible bioartist and educator. And they were thinking about biotechnology education, thinking about equity and access issues, and being like, why is the conversation not rooted in the voices of communities that have historically been excluded from science, from youth, and particularly when BioJam was started, with girls. And so they created a summer camp, so like an educational program that was for folks who have largely been excluded from science. We were thinking about this in a local context to the Bay Area around Stanford University. What are the communities who've not had a voice in the biotechnology development that happens at places like Stanford? And how do we shift the flow of knowledge? How do we shift the flow of resources to be grounded in community, and for which organizations like Stanford are a support? And so now BioJam largely serves the Salinas Valley. The Salinas Valley is about 2 hours south of Stanford, and it is a huge agricultural hub. So most of the lettuce and a lot of the fruits that are for sale in grocery stores in the United States actually come from the Salinas Valley and are picked by hand by agricultural workers there. A lot of those agricultural workers are immigrants from Mexico and other Latin American countries. And so we really wanted to highlight the knowledge and expertise of workers in this community that Rolando is from and that Corinne had been working in for many years. And so the focus of BioJam was to re-envision the future of bioengineering and biodesign, and through re-envisioning that future, just like we're doing on this call, reframing what it means to be a scientist and what science can be. So we wanted to start with art and culture and have that be the jumping off point for thinking about bioengineering. So how does this look? Well, the first thing that we always do is community need finding. What does the community want? What do the teens want? What do their families want? What are they interested in synthetic biology? And then how do we make that be the jumping off point for thinking about how synthetic biology could address issues in the community? And so the first thing we do is actually related to intergenerational knowledge. So much of the things that I've learned about biology came from my grandma. My grandma's was not trained as a scientist. She did not have any higher education, but she taught me so much about the natural world. And so we asked the teens to talk to their parents, their grandparents, their aunts and uncles, and ask them, like, what are some social justice or environmental justice concerns in your community? What do you guys care about? And the answers that we got back through these beautiful stories were things like well-paying jobs, having access to clean water, being exposed to toxic chemicals like pesticides, a lot of the things that we've already heard about here and that people are bringing up in the chat and earlier in this call. And so how can we use bioengineering in order to address real local problems, right? And the way we enter into that is creative exploration through art and culture. I remember when I was first getting involved in science, I thought it was the scariest thing ever. Like I was really intimidated. I didn't feel like it was for me. I felt like there was tons of words that I didn't know and that I'm not a knowledge holder in science at all. But what I did feel like I could do was draw, or I could be curious about the outside world, and I had creativity. And so we like to have that creativity and art and culture, things that young people already feel like they have expertise in, and use that as a grounding point for showing that no, those things are also science, and those are also grounding your expertise as a scientist as well. And so one example of an activity that we've been doing at BioGem for many years is focused on chinampas. So chinampas are a Mesoamerican system of agriculture that's similar to aquaponics systems. There's actually aquaponics lakes traditions and technologies around the world, but we're just highlighting Mexican ones. These are outside. These are chinampas that are outside of Mexico City because of the kind of cultural context of the community that we work with. And these are super amazing floating islands that use less water, that use less fertilizers, that are very biodiverse and produce large amounts of foods. And we were like, hey, could chinampas, a technology that's traditional, that's very culturally embedded in your community, actually address a lot of the environmental justice issues in your community in the United States, right? And they were like, oh my gosh, that's so cool. We always think about biotechnology as this thing that we have to learn or other people are teaching us, but actually we have the knowledge to develop biotechnologies ourselves. And how do we make sure that future biotechnologies not only serve our community, but are also rooted in community and traditional knowledge? And so this is one example. So the teens make these chinampas in a jar. We don't have the space, unfortunately, to build a giant floating island, but they make these chinampas in a jar. And I'm actually going to show you a TikTok that they made. You can see where they're making them. And we're using actually all the plants were donated by a local farm that we collaborate with. So I'm going to play the TikTok for you. It's really cute.
We can chase waterfalls and talk till all my hair dries. Just lie on the sand, each hand in hand in paradise. 'Cause we don't go long till we say so long.
It's so cute. The teens, when you teach in the classroom, so there's some students that finish really fast, and there's some students that are taking a little bit longer. So you can see how much I trust the teens. I handed them my phone, the students who finished earlier. I'm like, hey, you guys make a TikTok about what we're doing. So this is what they created for that day. um, oh, thanks, Diego. So the teens are developing, leadership and feeling like they're truly experts in biotechnology. So what do they do with that? Well, the teens get to show that leadership through teaching younger students. So we have all of these workshops with elementary school students where the students teach younger ones. And then we have these opportunities for them to give back to their community in other ways. So a couple of years ago, a teen actually built a full-size chinampa system at a local makerspace to engage the community. Jose Luis, you can see him here. And so sometimes the teens want to go on and actually share this as leaders and experts in their communities. Oftentimes we see young people as just learners, but young people are actually knowledge carriers, are actually oftentimes a way for getting other people, like elders in the community, to think differently through intergenerational dialogues. So we like to think about the camp, the educational program, is not just being one way. It's not just Stanford scientists teaching youth. It's actually youth and scientists working together to learn together. In fact, amplifying the knowledge of youth and providing opportunities for the youth to then be leaders, not just at a camp context, but in their own communities as well. And so the kind of focus of BioJam is aligned with the three sisters, La Milpa. This is a traditional growing practice centered in kind of the Americas that has three partners. It has corn, which is a structural component. It has beans that fix nitrogen in the soil and grow up the corn, and then squash that keeps the soil moist and limits weed production. And so if you could think about La Milpa as being like BioJam, we have the three sisters where we have community-led divisions. So we have an advisory board of 30 different community organizations that we work with and community educators that kind of shape the vision, just like the corn. They're the structural element. There's co-learning with youth. So these are the youth that, seed the soil through fixing nitrogen and grow up the stock. And then academics really is a supporter. So we're like the squash at the base side, making sure the soil stays moist and that there aren't too many weeds. So the idea is that the Stanford students and researchers are actually the supporters and not the leaders of growing biotechnology in community. And so I just wanted to shout out this is just some of the many hundreds of people who've been involved in organizing BioJam over the years through our youth, our advisory boards, our community educators, and our Stanford volunteers to make this happen. So I just wanted to share this story about BioJam as an alternative way of thinking about SynBio and biotechnology education that's really rooted in place, that's really rooted in youth knowledge and community and culture, as a way of thinking about not only making synthetic biology more accessible, but really reimagining synthetic biology through the lens of culture and community. And so the last story that I want to share is about empowering communities through local infrastructure. And so one question that we had when we were organizing BioJam is how do we grow more programs like this? It was actually really hard to organize BioJam because there's no place that has a lab that's near where the teens are. We couldn't have them driving 2 hours each way to get to Stanford to go to a lab. That was like not feasible at all. And so we're hacking together labs like in gardens and in classrooms, and we're trying to get spaces. So we were like, how do we get programs more like this? And how do we have spaces that are accessible to people to actually do science? And so Corinne had this beautiful quote, and she told me this. We were driving back from Salinas really late one night. It was like dark. I was sitting in the passenger seat, and we're talking about like how do we grow BioJam? And at first I was like, well, how about through schools? And Corinne's like, "Oh, schools are not accessible to a lot of people. How about libraries?" So she said, from years of work in schools, libraries, and community centers, I've seen how libraries, especially in under-resourced communities, and again in a United States context, offers consistent educators in spaces that nurture multi-generational sustained exploration. I believe that biodesign programming can build on that foundation, extending into library maker spaces as a natural community-rooted evolution in these trusted, inclusive settings. And so libraries, which are pretty common, I would say, in the United States, for kind of publicly accessible books. So people can go in there, get a book, check it out, bring it home, return it within a couple of weeks. And it's kind of a shared resource for book access that maybe we need to have something like this for science. And so this was the basis of this idea, libraries, just like libraries creating public access to books, that libraries might be these public spaces that created public access to science that could be in every community, that you could have these sorts of programs like BioJam that were very accessible, that were within walking distance to where people lived and worked. And that could be an expansion of educational infrastructure. Because right now, at least in my experience, I've always thought about education as going, you can start at home, then you go to school, and if you're lucky, you can go to university, and that's where you learn, right? But in reality, there's so many other places that people learn. People learn at art centers like I did, right? Community centers, like where you learn about kind of cultural experiences and things like that, and at places like the library. So what if we added in a LABrary into the mix? What if that was a place where people knew that they could go and learn, even if they didn't feel like they could go to school or they couldn't go to university? There was a space for everyone of all ages to go and engage in science. And so that was the fundamental idea behind this project, Biology for Everyone, that I was working with Drew Endy and David Kong with. We had an incredible advisory board, a huge team of undergraduate researchers, and many other hundreds of people who were involved in this project, including others who are on this call even. I just wanted to highlight quickly that everything I'm about to share is inside of this report that we published in late October last year, and a paper that just came out in Issues in Science and Technology just a couple of weeks ago. But I wanted to share this focus, library artistic reflection. Ooh, I want to hear more about what you're envisioning with that. I'm just looking at the chat right now. But we were really inspired by community biology labs. This is a little bit of an outdated picture, but there are these community spaces all over the world where people can engage in biotechnology right now. Some examples of the Floating Fab Lab in Peru that I mentioned earlier, Beno Juarez's efforts, Haquarium, which is based in Switzerland, and Tomorrow Lab in Thailand. which is a group of bio artists in Chiang Mai. And so one of the big gathering places for community bio is the Global Community Bio Summit, which I know several people, including ICE, have been involved in for many years. And I just wanted to highlight something that the Global Community Bio Summit made a couple of years ago, which was a shared statement of purpose. And I think that the shared statement of purpose does a really good job highlighting some of the goals of the community bio movement for folks who are not already involved in community bio labs.
Hola.
Madame.
Jambo.
Bonjour.
Hi.
Hey. Namaste.
Servus.
Vanakkam.
Привіт.
Hey everyone. Hello.
Kekwanu.
Our shared purpose
is to fundamentally
transform life sciences
And democratize
biotechnology
To inspire
creativity
and improve life
by organizing life science
Change makers.
and by enthusiasts
To build
an inclusive global network.
cultivate an accessible commons of knowledge
and resources, launch community labs
and projects,
And enable local educators.
To go out to the bang, woo! I'm gonna play some funk music for you now.
I love that shared vision and truly a global vision as well of community bio. And so that was where we were starting. I just wanted to give a couple more examples of what community bio labs do. A lot of the focus of this project was on the US, so I'm really eager to hear from you all about how this could be useful in a more of a global context, but I just am going to share a couple of US examples here. One is thinking about environmental science. It's something that people brought up in chat. The Baltimore Underground Science Base does DNA sequencing of the Chesapeake Bay, which was very polluted for many years because of kind of industrial work around that area, but they've been working to restore it. But they wanted to see how good the restoration is. So they've been doing DNA sequencing over many years to see what species have come back after restoration of the kind of toxic area. They also host an iGEM team, one of the best iGEM teams in the United States, the East Coast Bio Crew. You can see they're working here. BioCurious in Santa Clara has a focus on entrepreneurship. So they're a place where a lot of biotech startups that don't have a lot of money, but have an idea, a big idea, can go and work at a really, really, really low cost to develop out their company and their technologies. And GenSpace in Brooklyn, New York, really focuses on bioart and design. So how do we have spaces for artists who want to use biology but don't have access to labs to be able to go and create really a cutting edge art? And so these spaces, I'll use GenSpace as an example, can contribute to a local bioengineering ecosystem. They're not just islands on their own, but they can do things like work with local community organizations to engage the community, to have workshops. Like something that GenSpace sometimes does is they like go to local markets, for example, and present about what they're doing. They can engage with schools, colleges, and universities by offering kind of educational pathways, internships, and real world research experience when that might not be accessible to students. They can be incubators and help with science and technology innovation by offering research spaces, places for people to get jobs, start their companies. And finally, they can work with local, state, and federal governments to promote biosafety and biosecurity. When I was growing up, nobody really taught me how to be safe. You know, like we like to do a lot of canning, like we'd pick wild apples and put them in cans and store them for the winter time. And it was just my grandma who taught me about how to safely can so I didn't get botulism. There wasn't really a place that I could go to learn about that. So it could be one way to make sure that people knew about best public health practices and like other safety protocols with biology if there were spaces like libraries where people could go and learn. And one of the important components is just like a library has librarians, a library would need to have librarians. This is Desmond Goff, who's the lab manager of SynBio Lab in Seattle, Washington. And what librarians do is inspire passion for biology and then build community through the life sciences by developing programming in the spaces and also ensuring that the library is a safe and maintained environment. And so if your community doesn't have a library, which is most communities, the purpose of this project was to try to support the creation of more libraries. How do you envision a library from the ground up? And as you've already gathered from this talk, I love art. And so we created an art project. We created a really fun little diorama. It's a 3D, it's a printed PDF on a sheet of paper you can cut out and then just decorate with whatever you have on hand, with some markers, some colored pencils, and really draw out what you envision your library to be in your own community. And we worked with a video maker to actually create this really cute video. It's on our Instagram that I'm going to play for you now about how this works.
Hi, let me show you my LABrary. I envisioned a space where people are welcome to try biology or lab-related activities and hang out. First, I printed out the activity sheets from the Bio4ALL website. Then I started to think about how I wanted to design my LABrary. I remember thinking in high school that I wish we got more chances to explore and play with biology instead of memorizing concepts or tests. I also wish that I could share with my parents better how biology and technology can be accessible and interesting. And I also got their opinions on what they wanted to see in a LABrary. There would be a lot of hands-on ways to experience science through food, nature, and gardens. I love the idea of combining everyday science-related things with the culture of my community. Biology would be for everyone by having an accessible space with multiple languages where people will feel safe and excited to share their interests. And here's my LABrary with the representation of things and activities that I envisioned. It was an oddly moving exercise to try to create a space like this. I hope you'll have a chance to try it out too.
I love that Vicky included her dad. It was really wholesome. I'm like, yes, everyone's dad should be involved in designing the LABrary. That's great. Okay, so I have a question for folks in the audience, which was, if there was a biolab in your community, what would you want to do in it? Like, what would be cool to have in your LABrary? I'm gonna share about how other people have started libraries, but I would really love to hear maybe at the end of this ideas that folks have. I don't want to put anybody on the spot right now, but if you want to be thinking about that and put that in the chat, we can make it happen. So I would love to hear about that. There are kind of three places to start. First I was like, oh my God, if I was going to start a library, like I would be so overwhelmed. And the library like GenSpace that I was telling you about is what we call a big library. Like it's a standalone organization that has its own location that you can go into. But that's a lot to get your own organization, your own location, all the equipment and stuff. It might be easier to actually put it inside of something else. So you could make a medium-sized one that could be, for example, in a university. So, or you could make a little one at home, but I'll give you an example. So GenSpace is what we call a big library. So it's like its own completely standalone organization. You can go inside. Here's a picture of it. They have all the equipment. They do their own workshops, et cetera, et cetera. But a medium size is the BioArt Lab at the Chicago Art Institute. Andy Scarpelli runs this. It's within the university. It's within an art school actually, and they have a whole biology lab inside of the university that is for their students. That we can make one that's more publicly accessible, but that's like one example. And the third is what we call a little library or a little lab. So these are little cabinets that are, in this case, outside of Corinne's house in Honolulu. And in her little lab, she has Japanese kokedama moss balls, which are things that really people like in her community, and these like woven little baskets from local materials to really get people in her neighborhood talking about biomaterials and their love of biomaterials with things that are already accessible to them. And so little labs can be spaces to share community resources like kokedama, like woven pieces, things that people can share. They can build new relationships by introducing neighbors to each other and changing the relationship between, people and local plants, for example. And also sharing knowledge. Like people in the neighborhood may not have known how to make a kokedama or how to weave, but it's an opportunity to start sharing knowledge within the community. And this could look really differently. We have a network of little labs around the globe right now these are just some examples, Mumbai, India, Chiang Mai, Thailand, and Guatemala, and Mexico, and the United States. And so another question that I'm curious about is if folks were to build a little lab in their community, what would you want to find inside or what would you want to put inside? And the reason why I asked that question is because we're actually launching a second cohort of Little Labs right now. And the application ends tomorrow, actually, but if you apply on Monday, that's okay. This is a QR code. I can share this after the end of this talk. And everyone who is accepted into the second cohort will also get a bunch of free Foldscopes, which are these foldable paper microscopes to be able to distribute as well, because we have this partnership with Foldscope Instruments. So anyone who's excited about building a Little Lab in your community can apply now. And this is organized with a really amazing group, largely at the Fred Hutch Cancer Center and the University of Washington. And so just to wrap things up, in this talk, I wanted to share kind of how we might reimagine what science is through art and culture, how we might use education to center power in local knowledge, and then empower communities in that process through creating local accessible infrastructure for synthetic biology. So I hope that together we can make synthetic biology for everyone, and I really look forward to speaking with all of you in the Q&A. Thank you.
Thanks a lot, Callie. That's wonderful. So again, like back to like what Callie was just showing, like if you have any ideas on the LABrary that you want to share, just feel free to unmute, speak up, or just put into the chat. Other than that, if you have another question that was not related to what Callie just asked, like feel free to ask them as well. I think like all the the arts and science that Callie has shown in today's talk is very beautiful and also like very interesting at the same time. We are trying, like for the community ourselves, we were also trying to make sure that we make the education easy and accessible, and like combining the artistic style into our teaching is something that we wanted to do for sure.
Yeah, thank you, Ice. Well, really happy to have, I'm happy to stay on as long as people want to chat with questions or just ideas that you have. I would love to hear about your ideas. Also, feel free to reach out to me after this as well. Happy to be in touch. I can share my email in the chat if folks are interested in chatting more. But would really love to have this time to really hear from you.
I'm sorry if I have some discussion in the background, but I have a quick question.
Oh, you got muted, I think, Ice.
Sorry, I was trying to ask about the art and science part. Just wonder, like, is there any Little Labs or any projects that we are trying to combine the painting? I mean, you were showing the indigo project where you're trying to make indigoidine, which is a chemical that used to be isolated and then like made. But what about the natural paints? Like, is there any other things that we can try to leverage the various resources that we have in different communities to do some of the paintings? Because like that's one of the closest relationship between art and science, because all colors came from some scientific processes.
Yeah, so there's a couple of examples. One actually literally 2 days ago, I got an update about a low lab in the San Juan Islands in Washington State. And that low lab that's located in Friday Harbor Labs that you might be familiar with, ICE, is focused on cyanotyping. And so cyanotypes, if you've heard of blueprints, for example, it came from cyanotypes, which are a chemical, like a photoreactive chemical process whereby things that are put down onto a piece of paper that has the special cyanotyping chemicals, everything that's covered by the sun does not change color, but the rest of the paper that is exposed to sun does. And so this was actually the earliest type of photography. It was an early photographic technique that was used by, for example, botanists to take pictures of plants. And so a lot of like early seaweeds, for example, were cataloged using cyanotypes. And so one little lab that's in the San Juan Islands is also focused on cataloging local biodiversity using cyanotypes. And so the inside of that little lab is all the different materials for cyanotyping. And it encourages people to go out and actually forage inside of tide pools and to take records of what is there, the biodiversity in those tide pools in the San Juan Islands using cyanotypes. So that's not really a painting technique, but that's one example of an art-focused little lab. Another example is not a little lab, but is related. It's BioArtBot. So if anyone has used an Opentrons pipetting robot or heard of Opentrons, the BioArtBot actually has an online interface that you can plug in a little design, and that design, it's like on a 96 well plate format, will get pipetted by an Opentrons using a cloud lab to grow on an agar petri dish the picture that you created. And so there's a bunch of different, originally when Tim Dobbs made it, it was mostly different E. coli's that were producing different fluorescent proteins. Oh, thank you so much, Ice, for adding that. But they actually just did a new project where they were looking at trying to create or isolate different strains of mostly soil microbes or other environmental microbes from different places that were colorful that they could use instead. So creating agar art with locally sourced microbes, thinking about how we might work with our local environment in order to create new things or co-design new things, specifically related to pigments in microbes. So that's my second example. That's pretty cool.
Thanks a lot, Callie. Yeah, I mean, related to the BioArtBot, I think like there was a the one of the class and how to grow almost anything that was trying to use the Opentron to draw on agar as well. And this was pretty fun to try on because like when you have the robot, it can be very still and they can do like a much tinier drops than human hands can do. I mean, I tried to do some myself, and it takes a lot of training and trial and error to draw the proper paint using something that will just grow bigger. This is really
Yeah.
interesting thing to do. So
It's pretty fun. And like we did a workshop, we did a BioArtBot workshop like early like this spring. And so a bunch of the people who came because they were interested in doing art. But then the majority of the workshop was them just troubleshooting the Opentrons robot. And so they got like a pretty hardcore intro to robotics that they were not expecting from the workshop, but they really liked it. Like I think a bunch of moms who came for like a wine and sip kind of style art event like ended up getting really into robotics in ways that they were not originally anticipating. So all demographics, not just kids,
yeah,
adults.
it sounds very fun. I mean, like, and it's also like a very accessible robot setup that we can try as well. Right?
Yeah, not every day you get to play with robots.
Yeah.
Did you have something you wanted to share, Diego?
Hi, Kaylee. Nice to see you. Sorry, my internet died, and well, I am returning now.
Oh, it's good to see you. Thank you for being here.
And well, I have a question. I have a question because I want to conduct some workshops in communities here in the Andes, and my question is, based in your experience, if there isn't a community lab close to these communities after the workshops or activities, what has been the best way to stay in touch with them after the workshops, for example?
I think it's really important to have community partners that are based in the community. So like I work in Salinas because I can drive down there most weekends, right? And like teens will reach out to me and be like, hey, like things are not good, like I need this thing or whatever, like I'm interested in this, can you help me? And I can just like go there immediately. But I can't always be there. And so we have really deep partnerships with people who live in town with them in the same communities, are like a senior mentoring a freshman in high school or something like that. Or like community organizations that like provide snacks or like provide their transportation to school and stuff, or like seeing them all the time. And so I think it's really important to do this work where it's not like what they call parachute science, where you're just like going in and then leave, but like really are deeply embedded in the community for a long time. So I have a collaborative project right now with a undergraduate who's from Ayacucho, Peru. And so she's interested in doing community work there related to doing DNA soil sequencing with a nanopore DNA sequencer. And so she's actually working with her family. She told me, she was like, we just had a family meeting about the DNA sequencing project. that are all there, and they're like talking about what organizations they want to work with, and like who's there, and who's like who has the bandwidth, like what aunties and what uncles and stuff are just like around who like want to do it. So when she goes there, they do the workshops, but it's like part of the longer term engagement that's deeply connected with their family history over multiple generations and their family connections within the community. And I think that's really important. But the way we're going to do it is we bought a bento lab, and we have a nanopore. So bento lab has like it has like a gel electrophoresis rig and a like multi-speed centrifuge and a PCR machine kind of all built into it, and it's kind of like bento box sized. And so we're planning on doing a lot of that just like in community locations, and with the infrastructure needed, that would be physically there and not necessarily have to leave.
Okay,
But
great,
I'm
great.
curious.
And about your project with the undergraduate student, I will happy to help if you need any contact, or very happy to collaborate.
Yeah, okay, let's look. Okay, we'll have another call. See, this is awesome, you guys. Diego, yes.
Because
I'll reach out to you later.
because I am planning to do some workshops in the south of Peru, in Arequipa, and now I am in contact with some friends, biology friends in Arequipa. But Arequipa is close to Ayacucho too, and probably we can do something interesting together.
Yeah. Oh my gosh, how exciting. Okay, yeah, let's make it happen. She was actually in Peru a couple of years ago doing Foldscope workshops in Ayacucho as well. So she already has been doing some of this like science stuff, but it was not with the nanopore.
Perfect. Thank you, Callie.
Thank you. Oh my gosh. Thank you, Diego.
Yeah, I was really glad to hear that, we can initiate more collaboration through this kind of like workshop and webinar. So I think like I'm looking at the time, and I think like we're pretty good in terms of like the planned time, and maybe we can give Callie, give him some time to, move to the other commitment, because we all probably like have some, some plans around on Friday. With that, I would like to thank Callie again, who will share with us a lot of like, beautiful and interesting project in this webinar. The recording will be shared on our website. And if you are trying to, initiate any little lab, any library that you have ideas on, please feel free to connect with Callie or connect with us. And we just like, connect you guys together to think about something that could be cool to work together. I think that's it for today. And looking forward to see all of you again. We will have like more interesting webinar coming up. Right.
Wait, everyone, I'm gonna add into the chat the LAB cohort 2 link so you guys can all apply. Let's see here. I feel like, sorry, because I was giving the talk. I'm like, I can't get this link up. Give me 1 second. Okay, there you go. You can apply right there. It costs no money. We'll give you stuff. We also have some limited stipends available, like $50 to $100 probably, to build your little lab for some groups, if you're not based in the global north. So get your money, get your Foldscopes.
Sounds good. Thanks a lot, Callie. And with that, I think I'll just like stop the recording. See you guys again. Bye.